Thank you to the organizers for putting all of this together. I'm really delighted and frankly honored to be here and to have the opportunity to tell you all about some of our work today. I want to start by congratulating all of you for making it to the last scientific talk of the day. I realize I'm the only thing standing between you and the reception. So thanks for sticking it out and for being here. So I want to talk a little bit in general terms about the phage therapy work that my lab has become involved in over the last 5 years or so. But I guess I'll start by disclosing that nobody is paying me to be here. I don't receive any money other than the money that funds our research. So our funders are shown here. We're incredibly thankful to all of them, especially CFRI, for giving us the opportunity to do the work that we do. And to sort of outline what I want to cover today, I was going to talk about 3 different things. I'll tell you about the compassionate use phage therapy program that we've built over the last 5 years at the University of Pittsburgh, called the Pittsburgh Phage Program. And then to talk about the motivation for the New Horizons research grant that we received from CFRI. Take you through a single case, the very first case of phage therapy in a human patient that I was involved in, which was a person with CF that was suffering from a Burkholderia infection that we found phage for and gave phage to, and then had the opportunity to study the patient and try to understand a little bit what happened after we gave them phage therapy. And then how that has sort of driven, so the observations in the clinic from treating this patient have driven our research program in a couple of different directions. And so at the end, I want to tell you about how we've used lessons learned from this case of phage therapy to fuel research going on in our lab. Okay, so by way of background, usually when I go to a conference, I'm the only person talking about phage. I have the honor and I'm lucky that there've already been a couple of phage talks at the conference so far. But in terms of motivating the talk and by way of background, right, I'm interested in phage therapy because there's this pressing clinical need of antibiotic resistance. So antibiotic resistant bacterial infections are a looming threat to public health, not just in people with CF, but broadly. We have modern medicine has advanced so far, both in CF, but generally as well, to a point where we can keep people alive for so much longer. And that is amazing. A consequence of that, however, is that now we have lots of people that are severely immunocompromised and are uniquely vulnerable to becoming infected with bacteria, and they're not able to, fend off infections the same way that people with intact immune systems can. So we have these uniquely vulnerable populations of patients that become infected with bacteria, and then the bacteria can evolve, increasing resistance to antibiotics. So this is the sort of threat posed by antibiotic resistance. Hopefully you've listened to a phage talk to learn that bacteriophages are viruses that can infect bacteria. So the idea that the enemy of my enemy is my friend, right? So bacteria and resistant bacteria are our enemies. Phages are the enemies of those bacteria. So phages are our friends. And phage therapy seeks to use the lytic potential of these bacterial targeting viruses to treat bacterial infections. So we are trying to sort of harness natural evolution. The phages are all around us. They're in the air that we breathe, the food that we eat. We all are colonized. We're swimming in phages. We can't see them, but they are there. And they're seeking bacterial hosts to use as factories to make more of themselves. And phage therapy seeks to leverage this relationship for a therapeutic benefit. So we know that antibiotic resistance is a problem. We have this potential new tool in phages. So we've decided to put these 2 things together and to develop phage therapy as a new antibiotic approach for treating resistant infections. So that sounds amazing, right? Why can't we all just get some phages? So the harsh reality is that today, at least, there are no FDA-approved phage therapeutics for use in humans. And we're trying, but for patients these days, if you want to access this therapeutic approach, you have 2 options. So if you're eligible to enroll in a clinical trial, there are clinical trials seeking to test the safety and efficacy of phage therapy for a variety of different indications. But if you're not eligible for a clinical trial, another way that patients can seek phage therapy is through this thing called compassionate use. So compassionate use is for patients who we think could benefit from phage therapy, but they don't qualify for a clinical trial. And this is the way that we offer phage to patients at UPMC. As well as elsewhere, and it's called compassionate use. So this is literally one patient at a time, and I tell people that we are matchmakers. So we take bacteria from patients that have infections, and then we look for phages, and we're trying to match particular phages to a patient's infecting bacterial strain. And we've been doing this over the past 5 years or so at University of Pittsburgh. So we call ourselves the Pittsburgh Phage Program, or the P3, and this is how we organize our phage therapy efforts. So this is the P3 team. It's not just me. We have a whole crew of people. We have a website. We have an email address, phage@pitt.edu. The bottom right picture, Bree Bishop is our program manager. So if you email us at phage@pitt, Bree will get back to you and answer any questions you have. And I encourage anybody who's interested to reach out to learn more. And how it actually looks, the P3, is that we have both a preclinical pipeline as well as a clinical pipeline for phage therapy. On the preclinical side, we need phages. So to do phage therapy, you need phages. And when I started my lab at University of Pittsburgh in 2018, now 7 years ago, I had no phages. I was interested in phage therapy, but I had no phages. So I asked my physician colleagues to give me sort of like the worst of the worst bacteria. So I told them, think of a patient that had a really nasty infection. And if you had phage, you would have considered using it to treat that patient's infection, and then give me their bacteria, give me those bacteria. And so we took all these bacterial strains from my colleagues, and then we got some wastewater from a local wastewater treatment plant in the Pittsburgh area. And we started fishing in the wastewater using the bacterial strains from the infected patients as sort of our hook, our bait for fishing. And lo and behold, wastewater and sewage is teeming with bacteriophages. It's a gold mine for phage hunting. And so our preclinical pipeline starts with just looking for phages, and we mine a variety of different environments. So there's a lot of phages come from wastewater. We've also found phages in clinical specimens, so stool, sputum, swabs from patients with infections. There are phages in those specimens. And so we've been able to find lytic phages targeting some of our bacterial strains of interest from these sources. So with a lot of effort by trainees in the lab, we've taken sort of a one-by-one approach of fishing individual bacteriophages out of these sources. And then we isolate those phages and we characterize them to learn what are the species of bacteria that they target, what do their genomes encode, and based on the genome sequence, do we think it's going to be an appropriate phage that would be useful to use in phage therapy? And we've done that over and over and over again, and we now have species-specific libraries of phage. So in this schematic, you can imagine like each one of these vials is an individual phage in a library, and they might be grouped by the bacterial species that they target. So now we have a library of characterized phages. And then a doctor calls us and says, hey, I have a patient, they are really sick. Would you look for phages for them? Now we can take a bacterial isolate from that patient's infection and screen it against the library. So we do phage susceptibility testing to see if we can make a match. We're trying to match phages in our library to the bacteria from the patient. So we screen patient isolates, and if we make a match, then we tell the physician we have phage. Would you like to use phage therapy for your patient? And if they say yes, then we move the patient into our clinical phage pipeline. So once the physician says, yes, I want to use phage to treat the patient, then we have to make phage for that single individual patient. So we make patient-specific preparations. We biomanufacture phage in our research lab. We make a bunch of phage, we purify it, and then we formulate it either on its own or mix phages together into a cocktail. So I also tell people we're like mixologists making phage cocktails of different flavors and different varieties for people. Once we formulate a phage cocktail for a patient, we have to test it, right? So we've basically used bacteria to grow a bunch of phage, and we've purified the phage away from all the other stuff in that that the bacteria have, but we have to test the cocktail that we formulated to make sure that we think it's going to be safe if we give it to the patient. So we test sterility, we test for endotoxin, we test for exotoxins to make sure that all of these impurities are at a sufficiently low level that we don't think it's going to harm the patient if we give them the phage that we've made for them. After all of that testing, we have to get approval and consent. So we apply to the FDA for a single patient IND. And if the FDA says okay, then we ask the local IRB at the hospital where the patient's being treated if they approve, and we get their approval. And then finally, we ask the patient to consent. Phage therapy in this mechanism is an experimental treatment. So the patient has to be willing to assume a little bit of risk. And that's in exchange for a potential benefit of receiving this therapy. So after all of that approval and consent process, then we can initiate treatment. And of course, we monitor the patient very closely to make sure that the therapy is safe, that it's well tolerated. And then ultimately, we would like the patient to receive a clinical benefit from getting phage therapy. Okay, so this is a time and labor intensive process, but right now it's the only way that patients can access phage therapy outside of a clinical trial in the United States. So we've been doing this, like I said, for the past 5 years or so, and this is just a breakdown of our volume and what we've accomplished. So the flow chart on the left is just showing from 278 initial requests for screening for phage therapy, we were able to identify active phages for about 70% of the patients that were referred to us. And then that boiled down to 48 patients, or about 20%, that were actually treated with phage therapy. And I had to turn in the slides a week ago, and they're already out of date. So just in the week since making the slides, we've gotten FDA approval for 2 additional patients. So we're actually now up to 50 patients that we've gotten permission from the FDA to treat with phage therapy. The map on the right is showing for 2023 and 2024, all of the dots are different places in the country where we've received requests for phage susceptibility testing. So the big dot in Western Pennsylvania is Pittsburgh. A lot of our business obviously comes from our local health system, but we are a nationwide program. Actually, just last week, we received isolates from a person with CF that actually lives in Alaska. We got our first Alaskan patient sending us isolates to test for phage susceptibility. And then the pie chart on the bottom is showing the distribution of bacterial species that physicians are requesting phage therapy for. So it's pretty striking, at least to me, that almost half of all the requests that we get are for Pseudomonas aeruginosa infections. And I think this reflects the sort of clinical burden of drug-resistant Pseudomonas infections in a variety of patients, including people with CF. And you can see some other, prominent CF pathogens like Burkholderia and Achromobacter and Steno are also in the pie chart as well. We are not restricted by disease state. We serve anybody as long as we have phages for their pathogen. So if you look at the sort of flow, the numbers, you might notice that we only find phage for about 70% of the isolates that we screen. And it turns out that making a phage match, or the likelihood that we can find active phage, varies quite a bit depending on the pathogen that we're screening. So in the case of Pseudomonas aeruginosa, almost 75% of the time when we receive an isolate, we can find active phage that are able to kill the bacteria. For Burkholderia, though, that ratio is flipped. So only about 25% of the time when we receive a Burkholderia isolate to screen, we're able to find phage for it. And I'll talk about that a lot more in the next couple of slides. And then for a pathogen like Enterococcus faecium, it's about a flip of a coin, 50-50, whether we're going to have an active phage in our library that could target that strain. Okay, so this is, to me, it's an interesting question. Why is it so hard to find active phages for Burkholderia? It's really striking. Like, we have the hardest time of all the pathogens that we look for phage for. Burkholderia is like persistently difficult to find active phage. And there are a couple of reasons we think this might be. The first is that Burkholderia is an environmental microbe. It doesn't naturally live in people and colonize people. Its preferred environment is actually outside in the soil. And in the soil, bacteria, to survive there, need to be able to withstand starvation and desiccation. They need to be able to fight with other bacteria around them. So Burkholderia seems to have this natural hardiness, and is also naturally quite resistant to a lot of traditional antimicrobials. Another reason why we think it's hard to find phage for Burkholderia is that the genomes of these bacteria are very large, and the bacteria themselves are very diverse. And then, so the red circles on the right, it's a little hard to pick out with the projector, but these are different bacterial strains that all belong to the same Burkholderia genus called Burkholderia thailandensis. And each picture is a different bacterial strain. And if you look at these are bacterial colonies that are growing on a Petri dish. And hopefully you can appreciate that each each colony looks a little bit different. So you have some colonies, some strains that are really kind of wrinkly, and then others that are very smooth, and each one looks a little bit different. And this reflects that the genomes are super diverse. The bacteria themselves can do a lot of different things. And from a phage perspective, it's almost like you need a different phage for every single one of these strains. And then another reason why we think it's difficult to find phage for Burkholderia is that the genomes of the bacteria themselves encode these things called antiphage defense systems. So these are like the immune system of bacteria. They are systems that the bacteria have to be able to resist phage infection. And when we look in the genomes of Burkholderia strains, we find a lot of antiphage defense systems there. And then finally, they're just simply understudied. So not a lot of people care. And there's not a lot of attention or resources for studying Burkholderia. So all of these things we think combine together to result in making it very difficult for us to find phages to target Burk infections. So I'm going to switch gears a little bit now and talk about a clinical case. So this was my entry into phage therapy. So it all started in the fall of 2020. Excuse me, I'm going to take a sip of water. So I got to Pittsburgh in 2018, and I spent about 2 years telling all my colleagues that I was interested in phage and asking for strains and hunting for phages and just like telling people I was interested in phage therapy. And it took about 2 years to actually get to treating a patient with phage. But this was a 32-year-old woman with cystic fibrosis that was known to be colonized with a Burkholderia species called Burkholderia multivarians. And she had been colonized with this bug for quite a while, almost 2 decades. And she received a double lung transplant in 2017, but unfortunately in the fall of 2020, she developed pneumonia and also sepsis with this strain of Burkholderia that she had been colonized, for a very, very long time with. So this is a case report that is published, so you can read the paper for all of the nitty-gritty details. Unfortunately, it's a sad story. She ultimately succumbed to her infection, but it was the opportunity to look for phage for her, to try and find phage, to get a phage for her, give it to her, and then learn quite a bit about giving phage to patients was for me like an extremely fulfilling and helpful experience. So the treatment course is shown here. It's quite busy, but there's a timeline in terms of a day that we made an observation. Above the timeline are blood cultures or respiratory tract cultures that were either negative in green or that grew Burkholderia multivarians, which is all the boxes in red. Hopefully you can appreciate, if you look at the antibiotic therapy, all of the different antibiotics that she was on, trying to sort of I liken it to like a forest fire, like all of the different fire departments trying to put out the forest fire of her raging Burkholderia multivarians infection. And you can see, despite all of these antibiotics, her blood cultures and respiratory tract cultures were still growing this organism. And that's, it's just staggering the level of resistance that some of these bacterial strains are capable of. Okay, so down here on the bottom is the timeline for finding phage for her. So we initiated a phage search, and miraculously, somehow 10 days later, we were able to start her on phage therapy. This is actually the fastest that we ever found phage for a patient. And it's actually entirely due to Ben Chan, who spoke this morning. So we sent her isolates to Ben at Yale, and he found a phage, grew that phage on her strain, made enough phage to be able to give her treatment with phage therapy, tested it sufficiently that we could satisfy the FDA, and then the FDA agreed to let us give phage to the patient under emergency use. And 10 days after the search went out, Ben, I think, was working through the night to find and make phage for this patient. So the patient received about a week of phage therapy through their they were ventilated, so they got it through the respiratory tract, inhaled phage therapy, and then they also received a single dose of IV phage before unfortunately succumbing to their infection. So this patient's family was willing to consent to let us collect biospecimens from the patient, both before and during their phage therapy. And we were able to take those biospecimens and bring them into the laboratory. And we looked using quantitative real-time PCR to assess how much bacteria was in the respiratory tract of the patient. And then we also looked for phage in her respiratory specimens. So this is showing the bacterial abundance over time time. On the x-axis is the day post-phage initiation. So she was receiving phage every day during this time. And we can see if we look at the abundance of bacteria in her respiratory tract samples, we see a small but significant decrease in the abundance of the bacteria in her airway over time. And what was perhaps more interesting to us was when we looked at the phage. So the phage that she received was a single phage called BCH7. And we looked for DNA corresponding to the phage in her respiratory tract specimens. And we didn't find any phage for the first 5 days, even though she was receiving inhaled phage therapy. But on day 6, all of a sudden we saw a spike, and we were suddenly able to detect the phage in her respiratory tract. And we have ideas about why this might be. It's possible that it took this long, it took 6 days for the phage to actually start to replicate and reach a high enough level that we could actually detect them. We think it's also possible that the way that she was receiving phage through nebulization may have been destroying the phage particles. And so we're not actually sure how much active phage we were delivering to her airway. And so again, it may have taken a while to get enough phage into her airway for the phage to actually be able to do anything. This nonetheless looks to us like a microbiologic response to phage therapy. Despite not finding phage at the beginning, we were able to detect it by the end, which we thought suggested to us that this phage that we had identified in the laboratory as being active, when we gave it to the patient, we could see evidence that the phage was actually replicating in the patient. We also had accessible to us a variety of bacterial isolates from this patient, going back to the first couple of isolates collected from her in the early 2000s. So we sequenced the genomes of about 15 different bacterial isolates from her and confirmed that the bacterial strain she was infected with, all of the isolates were closely related to one another. So on the far left of this figure is a phylogenetic tree where we plot the bacterial isolates relative to one another, comparing their genome sequences. And there's some diversity, right? The bacterial isolates are not all exactly the same, but compared to Burkholderia multivorans from other patients, the bacteria from this patient were all very closely related to one another. And the rows here, each row here is an isolate, and the columns are different antibiotics. And we've coded the susceptibility as either susceptible, intermediate, or resistant. And what we notice is that the earlier isolates that we got from the patient in the early 2000s were generally susceptible to antibiotics that are frequently used to treat Burk infections. The final column here is the phage that the patient received for phage therapy. And what was very interesting to us was that the earliest isolates from this patient were actually resistant to the phage that she received for phage therapy. It wasn't until sometime later, about a decade after she was initially colonized with this strain, that the isolates became susceptible to the phage that she was ultimately treated with. And that activity is summarized here. So on the left here is an early isolate. We've spotted dilutions of phage on top of a bacterial lawn of the isolate, and there's no evidence of phage inhibition of bacterial growth. But in the later isolate, that was collected closer to when she was treated with phage therapy, now the isolate actually is susceptible to the phage that was used for therapy. And if we drill down a little bit further, we can see that this is likely due to changes on the bacterial surface. So this is the lipopolysaccharide from those two different bacterial strains. And there's a nice O antigen banding pattern here in the phage resistant strain that's absent in the phage susceptible strain. So we thought it was very interesting that over time, this patient's isolates evolved to become more antibiotic resistant, but also that had a trade-off and they became phage susceptible. It also raises questions about what is the right time to screen patients for phage susceptibility. How long can you guarantee that a patient's isolates might remain susceptible to a phage that you would want to treat them with? But nonetheless, we learned an incredible amount from this case. And to summarize just two main lessons that we learned, the first is that it's incredibly difficult to source Burkholderia targeting phages, especially when you have short notice. And that the second lesson was that phage therapy might have been more effective for this patient if we could have optimized the activity, found better phages for them, and also we might have been able to improve the phage delivery. At the time, we simply didn't have the time. We were sort of, sprinting to try and give anything to the patient that we could. And, now in hindsight and reflecting, we think that there are ways that we could have improved our process. And that's what we're focusing on now on the research side. So to tackle the first lesson, so we know that we need more Burkholderia targeting phages. It's been very difficult to find active phages against Burkholderia isolates from people with CF. At the same time, at our local center, we've collected and sequenced a lot of Burkholderia clinical isolates from patients. And when we look in the genomes of these isolates, we notice that they encode a lot of prophages. So prophages are bacteriophages that infect bacterial cells, and rather than killing the cells and lysing them, the phage integrates into the bacterial genome and becomes what we call a prophage. And these phages are replicated along with the bacteria, and they basically hitch a ride as the bacteria divide and grow. So these prophages are sort of the remnants of successful phage infection. They're not immediately useful to us because they're not going to kill the bacteria the way we want them to, but we wonder if they might be a useful starting point for example, engineering lytic phages. So we wonder if prophages from one Burkholderia clinical isolate could lyse a different bacterial strain. So we wondered if these prophages that are present and sort of abundant in Burkholderia clinical isolate genomes could be weaponized to kill other bacterial isolates. And to do this work, we relied on some collaborators. Lee Harrison is another professor at Pitt, and Katie, Marissa, and Vatsala are some folks in his lab. And this work was the master's thesis project for a trainee in my lab, Haley Nordstrom, that I'm going to tell you a little bit about. So I've been talking, I mentioned already the idea of lytic phages versus prophages. So phages have two different lifestyles. Lytic phages will simply use a bacterial cell as a factory to make more of themselves, and then ultimately lyse the bacterial cell. And those lytic phages are the phages we'd like to use for phage therapy. This lysogenic cycle is when the phage integrates into the bacterial genome and then hitches a ride and is replicated as the bacterial cell replicates. And so we want to know if these prophages could be a useful starting point for engineering of lytic phages. So the way that Haley approached this project was to induce prophage release from bacterial cultures. So she used a chemical called mitomycin C, and she enticed the bacterial cells to liberate their prophages. And then she took a filtered supernatant and then spotted it on other Burkholderia strains and looked for evidence of phage infection. So she did this in a sort of 32 strain by 32 screen. So she screened 1,024 pairwise combinations of source lysates and target isolates. She only found 11 pairings that showed evidence of phage activity or a 1% hit rate, but that was better than nothing. And she pick passaged and isolated 7 different phages, 4 of which ended up being non-redundant with one another. And after doing all of that screening, she was still smiling at the end, as evidenced here. I found her smiling in the lab. So we actually didn't know if anything was going to come out of it. So the fact that we got 4 new phages out of this effort, where we didn't use any wastewater, we just took strains and sort of found phages that were active in their genomes. Okay, so we took 6, so the 4 phages Haley isolated, and then 2 other phages, including BCH7, which was our clinical phage used to treat our patient. And she screened them against 35 different Burkholderia clinical isolates, spanning lots of different species of Burkholderia. And what we found was that most of the phages in this panel were active against multiple different species, which was surprising to us. We thought phages would be very specific and only target one particular strain, but we saw a lot of sort of cross reactivity here. We identified one of these prophage-derived phages called Bcc-02 that had greater activity. It could basically target more strains than any other of the phages. And we only found 8 isolates here that were resistant to all of the phages that we tested. And interestingly, all of those isolates were Burkholderia multivarians. And this has been a common theme in our work. We don't really understand why, but as a species, Burkholderia multivarians seems to be more resistant to phage infection. And we don't understand why, but we think that it's potentially interesting. So this was just with Burkholderia isolates from clinical sources, sort of all comers. We wondered specifically about would it look the same? Would the data look the same if we screened just Burkholderia from people with CF? And that's what we wrote a grant about and received the New Horizons Award to study. So we took most of these same phages and screened them against 100 different Burkholderia clinical isolates, now only from people with CF. So it was 2 isolates each in 50 patients, so 100 isolates total. And we're thankful to John LaPuma and Lindsay Caverly at Michigan for collecting these isolates, for saving them, and for making them available to us. This sort of one graphic summarizes lots of work by people in the lab to screen all 100 isolates against the 6 different phages. But this is an activity matrix. So every row is a phage that was tested, and then every column is an isolate. And they're colored in the Michigan colors of yellow for active and blue for inactive. So yellow is good and blue is bad in this screen. And hopefully you can appreciate, as other than just being visually pretty to look at, right? There's a lot of variability between the different phages and the different isolates. So we found some interesting patterns of activity when we broke down this data both by phage and by species, and I'll show you those breakdowns now. So if we think about the first focus on the phages, so which phage was the best? Interestingly, it looks like BCC02 was the most active phage. It was able to target the most number of isolates in this data set. And then we also saw interesting patterns of phage susceptibility by different species. And we found that, for example, B. multivorans B. multivorans isolates were susceptible to fewer phages, whereas B. orbicola isolates seem to be much more phage susceptible as a species, which was interesting. And then the other thing that this data allowed us to do is think about pairs of isolates from the same patient. And actually, Dr. Whiteson showed this paper yesterday. So I'm going to have to tell John LaPuma that his paper got like 2 shout outs at this meeting. He'll be thrilled. But we found that only about half of the patients that were tested had similar susceptibility profile between the 2 isolates that were tested. So half of the time when we got 2 isolates from the patient, they had different susceptibility profiles. And this has important bearing on sort of using phage for, how you pick phage and how you design a cocktail for a patient. Okay, and then the last thing to talk about is the nebulization and the aerosolization. So for patients with respiratory tract colonization or infection, right, we would like to deliver phage locally to the site of infection. And for the respiratory tract, this means aerosolization or nebulization. So prior studies have suggested that the simple fact of nebulization destroys phage particles. And we wondered how might this affect Burkholderia targeting phages. So we tested 3 different nebulizer types. So 2 of them are jet nebulizers, and one of them is a vibrating mesh nebulizer, and they're shown here with their brand names. And we used, again, the BCH7 phage, the phage that we had used clinically to treat a patient. And we start with some amount of phage as input, and then we nebulize the phage, and then we quantify how many active phage particles are still there post-nebulization. And, shown on this scale, it looks like they're not that bad, right? From the input of 10 to the 8 to 10 to the 9th viral particles, we only see about a log, maybe 2 logs different depending on the nebulizer type. And that's encouraging, but the flip side is if you think about the percentage of viability that's lost here, a 1 log drop in phage titer is a 90% drop in viability. A 2 log drop in phage titer is a 99% drop in phage viability. And so it's helpful to look at the data like this as well. And from this one experiment with 1 phage, our conclusion is that the Aerogen vibrating mesh nebulizer is actually the least bad, right? It only inactivates 80% of the phage particles, whereas an AirLife is inactivating like 95% of the phage particles. But we think this is an important consideration and something we need to think about in sort of developing phage therapy as an inhaled medicine for patients going forward. So I'm gonna stop there and conclude with the idea that phage therapy targeting Burkholderia is available for people with CF, but it's only available on a single patient or compassionate use basis. There are clinical trials that are planned for Burkholderia specifically, as well as broader phage therapy clinical trials for a variety of different organisms. We think that for Burkholderia, prophages are a potentially useful source of phages, but you have to do some more work. We've identified candidates for further study and development, such as genetic engineering. And in future directions, and with continued support from CFRI, we're moving into susceptibility testing with more phages, with collaborators at Texas A&M and the Israeli Phage Therapy Center, as well as nebulizer testing and phage antibiotic synergy testing. So I'm going to stop there and thank all the folks in my lab, many of whom did the work and put in the time so that I could come and tell you about this. I'll leave you with a nighttime view of Pittsburgh. It's just about nighttime there now. My body is still on the East Coast. And I thank you all for your attention, and I'm happy to take questions.